मैडम सार एपर्यंत आसी नांती
हेलो हाँ सर मु फोन कर दूँ गोटे सेकंड
मैडम सार एपर्यंत आसी नांती
हेलो
मैडम ठीक है
Good afternoon, one and all. We'll soon be starting our session. Sorry for the delay, participants. We'll soon be starting our series. Due to a technical snag, our speaker is unable to join. He'll be joining within 2 to 3 minutes. So kindly bear with us.
No, sir, is available.
So we have our guest with us. Once again, a very warm welcome to one and all present today. It's our fifth technical session today of the webinar series that is Kindling Curiosity, Ushering Young Minds into Research, organized by Rajdhani College, Department of Geology, in collaboration with Keys deemed to be University. Without any further delay, I would now request Dr. Banditarat to kindly present the address to our keynote speaker of the day. Madam, please.
I'm honored to introduce our speaker of today's session, Professor Jagneswar Dandapat, Professor and Head of the Department of Biotechnology at Utkal University, Odisha. In addition to his current position, he is also a coordinator for Department of Biotechnology, Government of India supported teaching program. Additionally, he serves as a coordinator of World Bank and Department of Higher Education, a Government of Odisha sponsored Center of Excellence in Integrated Omics and Computational Biology. Professor Dandapat obtained his BSc, MSc, MPhil, and PhD degrees in geology from the same university that he currently serves. Professor Dandapat is an expert in the field of cell biology and immunology. Prior to his tenure at Utkal University, he has held the position of lecturer at North Odisha University. His research group addresses questions related to redox regulation, epigenomics, and proteomics, focused on thyroid disorder and neurodegeneration in a variety of models ranging from silkworm to rat. Professor Dandapat has over 40 publications in international journals of repute and has guided many PhD and MPhil scholars. An avid researcher, he has been invited to present his work at many conferences and seminars. Professor Dandapat has also authored and co-authored several books focused on training manuals and prawn culture, among others. An eminent scientist in his field, Professor Dandapat holds prestigious membership in several national level academic committees and policy-making bodies. He has been conferred with many awards, honors, fellowship, and patents too. His group is engaged in collaborative research with various national and international institutions like ILS, NISA, RPRC, IMMT in Bhubaneswar, NCCS Pune, and Johns Hopkins University and Mississippi State University in the USA. We are extremely fortunate to have Professor Dandapat with us today to share his thought on doing science for profession, passion, or career. Sir, thank you so much, sir. The platform is all yours, sir. Thank you.
Thanks. Yeah, thank you, madam. Am I audible?
Yes, sir. Yes, sir.
Okay. So before I start, I beg apology, just having a delay because of some network problem. Let me start my talk on today's and on this great event. And before I start my talk, I must express my heartfelt gratitude to the organizer taking such an important initiative to kindle the curiosity among the young minds. So respected Principal Madam, Rajdhani College, Dr. Sashmita Sharangi, Coordinator, Case, Dr. Rashmita Mahapatra, organizer, Madam Dr. Bandita Rath from Department of Zoology, who has taken a lot of pain to initiate this program, and Dr. Priyadashini Rautrai, Lecture Department of Zoology, who is the moderator of this program. So I must congratulate all of you taking such a nice initiative in the state and to organize this national webinar. So my topic of today's discussion under the umbrella of this kindling curiosity and to ignite the mind of young to have the passion, to develop the passion, to become more passionate in the research. So I've chosen a topic, doing science for profession, passion, or career. So the topic is definitely really challenging for me because it's a lot of things that I have to balance, and particularly audience or on this virtual platform, the heterogeneous people starting from some of the students, graduate students, postgraduate students, some research scholars, even some faculty members, as I heard and as I observed in the last few meetings. Now coming to my topic directly, before I start the formal lecture, I must confess few things that during my course of lecture, I'll be using some of the examples, I'll give some of the cases, I will cite some of the cases, and none of this example or these cases is never intended or intention to promote any individual, to promote any organization. Rather, my sole objective to cite those examples, include those things, including some of these informations from my own department or lab, that is only to ignite the young minds, to inspire the people, particularly the students, the focus group, for doing science. So from the beginning of my talk, I make this stand clear. So coming to this doing science. So why people should be interested in science? One simple question for that. Science contribute for a better world. How the science contribute for a better world? What is the scope of science? What are the different career options for science? What type of professions one can choose if he or she wishes to do science, and how she can or he can achieve these things during one's career, and what type of options are available, what type of professions one can adopt during the course of his life. So what science is that I have given one line, that science contribute for a better world. Now, if I say that science is nothing but all about innovations, it is about the discovery, search, and research. The people having the scientific bent of mind do the things, try the things, do experiment, try to make some breakthrough. Scientists, they spend time silently in the laboratory. Scientists are crazy sometimes, having a crazy brain, and they are after the discovery of many fascinating things. Of course, for the scientist, for the persons who opt the career to do science, there are a lot of fascinating career disciplines are available that I will discuss one by one. So science all about doing things that nobody else has ever done before. This is what you see. It is exciting, incredible. Allow us to understand the nature of things. It leads to radical innovations. Of course, it has immense possibilities, and it has the ability, or it has the power to revolutionize the world. If I say the sky is the limit, even not applicable to explain career in science, such a career option is available with the people who are interested to do science. But I must say, to follow a career path, you are most likely going to have a highly logical, rational, and most importantly, you must be having you must have a deep passion about to choose your subject. And I will definitely give some examples throughout my deliberations. I will focus on three things during my lecture. What are the career options available? Some of the speakers have earlier told. What are the professions one can adopt, and what are the opportunities, and how the passion is more important to decide and to make your journey successful, either in your career or in your profession. I will ask three important aspects to cover. Research is an integral part of every scientific career. That, let me tell you, research is an integral part of every scientific career. Product and process development is another outcome of science. And if you really want to do all those things, you want to go for it, then you should have a proper planning in your life. In a recent survey by Nature, it's very, you know that one of the very reputed journal in 2018 made a survey on more than 6,000 people, and most of the respondents, around 70% or more of the respondents, they expressed their satisfaction that those who are in different profession in the science, or they have opted career in science, they are satisfied with respect to their profession, their job profile, their salary, and other things. But still, that I don't understand. Sometimes the people, they don't show enough interest to make their career, to enter into the science, to choose their profession or career in science. So let us see at the end of this talk that what are the immense possibilities we have if we decide to choose science in a career or a profession. And of course, what we have to do, we must do with full passion. So you say that we are really interested to do science, we're wondering to take a career in science, then what next? Science as a career? Coming to that, I must tell one thing, that every mind is creative, every mind is inquisitive. So if you wish to enter in this area of your profession or your career with a scientific bent of mind, that you must acknowledge that I have born with intelligence. God has given me enough rational thinking, and I have enough opportunity in the world, above the earth, under the earth. So every mind is creative, every mind is inquisitive. So what are the options? So I'm not going to discuss much of it, because some of earlier speakers have already explained all those things, and it is also available. And being a student of science, I think all of us should understand that we have the different disciplines available with us: the natural science, that life science, the physical science, the medical science, agricultural science, and we have n number of avenues even after +2, even after graduations. So these are some of the examples I can tell, that these are some of the disciplines, subject-specific disciplines, some of the profession and some of the career. And it's not like that long ago there are a few options are available for Asian students. That is not the case now. We have ample avenues, we have ample possibilities with lot of enthusiastic professions, and you will get utmost satisfaction by virtue of your doing the things. Now, how we can go to our next step? Once a student opt for the science, that or think that he should choose or she should choose for the science career, we have number of avenues. I must realize that what is my strength, what is my passion. If I understand that this is my passion, I must explore the possibilities, what are the avenues left with me. And if I know my strength, if I know that possibilities, then I should choose what I like the most. And if I choose that what I like the most, I should work with deep passion, and I will make it happen. Then once I decide that I will choose my career in any of the professions related to the science, and I must have a deep passion to do all those things. What next? Before I say what next, that we have ample avenues. So when I start my journey of my career as a scientist, as a teacher or academicians or even other professions, for everything, my learning is purposeful. And when the learning is purposeful, creativity blossoms. When creativity blossoms, thinking emanates. And when thinking emanates, knowledge is fully laid. When knowledge is laid, economy flourishes. This is what I have told you, that science for the prosperity of the world, for the prosperity of the better tomorrow for the society. So science has the possibility, enough possibilities for the development of nation, for the development of a globe. Now, once a student complete his formal educations, make himself or herself eligible, n number of fellowships are available, n number of fundings are available from national and international resources. So fundings is not a problem if I have a desire to do. So there are a list of funding agencies I have given, and is also available in all public domain. And when only things that I have to decide that what I should do, where I should do, how I should do. Now, when you have a funding agency available to you to support your fundings, and you can do a formal research as a part of integral part of doing science with a fundings or with a fellowship, because without fellowship I can't do anything. I cannot move ahead because I need to fill my stomach. Now I have a number of professions around me. Being a science, being a career, if I have a career in science, I have ample avenues starting from academia, industry, and so many things, and many avenues are available with me. Again, to supplement my earlier slide, that is again good number of options, not only academy, not only industry, not only research, not only medical or agriculture scientist. I have many options. So I have a research bent of mind. I want to make my career in science. I want to do progress in science through my profession or career, and I have number of options available with me. So I should not think, I should not give a second thought that why I am entering. I have decided I will enter, and I will do it, and I will make it happen. So these are some of the examples that you have before you. You can be a forensic scientist, you are an environmental scientist, you can be biochemist, you can be biophysics, you can opt medicine, you can start an industry of food and cosmetic industries, pharmaceutical, patent officials, educations, so many things. So many different domains, disciplines are available. And because I have a number of avenues, number of professions before me, and all are equally charming, all are equally important for the building of the nation, for the building of your career. Now, for every career, for every profession, if you want to make it a success, if you want to get the full bloom of it, you need the proper guidance, a guided spirit. You need a proper platform, and you must have a value addition to your existing idea, existing knowledge, and existing inherent strength. You have an inherent strength with you. So there will be value addition in the right time, and the right kind of value addition that will move you, or that will help you to land with prosperity. So we have human resources, you have a human mind, and I need a value addition through a right kind of platform, right kind of inputs. Definitely it will culminate good success, lots of achievements, and I'll get skilled human resources with global competence. Skilled human resources will global competence will definitely is a welcome step for everyone for building the nation to contribute in the better world for the tomorrow. I will give you an example that how value addition is important. I will cite an example of my own department, Department of Biotechnology at Utkal University, which is supported by Department of Biotechnology, Government of India. So everywhere we get students from different backgrounds to study only with an objective to study master's program in biotechnology. So we get the students from different disciplines, from different race, different ethnic population, different states, having different aptitude, but different strength of learning, but they have a vision, they have one mission that they do want to study master's program, they want to do science. And that is the right kind of value additions through human skill, human resources, through infrastructure, through moral support, and overall guidance, spirited guidance. And with these inputs, that learning outcomes, it comes out, they do success. They exhibit success in different national level examination. That is the entry to start their career in science. And that has been proved since last 15 to 20 years that every year you get the students from the different inherent strength, and with the right kind of inputs, the students come out with flying colors. We have tested. So this is I can say, a case study. So learning needs freedom to think and freedom to imagine, and both have to be facilitated by a dedicated teacher. Now coming to this research, I have told you that research is an integral component of this doing science. Research is definitely an integral component of doing science. Without research, that science cannot progress, science cannot move. So search and research. So research is not always a linear relation. I should not expect I am getting this one and to this, next to this, I will get this one. So it's not always a linear calculation or a linear relation. A good researcher sometimes pips or sometimes think to pip through a new window to establish a novel linkage at the crossroads. We do not know that what will be my discovery, what will be my finding, what will be my unraveling the things from my experiment. So it is all depends on my experimental plan, my knowledge, existing knowledge, my design of the experiment, and my objectives, and how sincerely I am doing all those things. So it is the process. I've told that research is a process, and it is the process of innovation that knowledge is converted into the wealth. So a good research, a planned research definitely leads to innovation, and that innovation is transformed into wealth through our knowledge. Coming to this, once I have my entry in the research to exhibit or to pursue my career in science, I must understand that what research is. So creativity is saying the same thing as everybody else, but thinking of something different. This is what is again another definition of research. So creativity, that science, that research might be done by most of the people, but they have not thought about this. So thinking in a different way, thinking differently with my findings, with a rational thinking, rational thought process, that can be a new story, that can be a new knowledge. So research leads to a finding answer to number of questions, and it is definitely systematic search for a truth, search for a knowledge, and it is definitely a systematic search for information on a specific topic. So research sometimes may be very focused, with focused objectives. It should give answer to number of questions: what, why, when, how, where, and who. So with this, that we have to answer a lot of questions during my journey in research, and research is an integral part of science. Now, if I formulate a research, if I want to do research, I have options that I'm going to do a basic research, or I'm going to apply research, or the combination of both basic and applied research. For examples, I can tell you that I have an information with me that, or that information is available with me, that caffeine ingestion improve calcium bind with troponin. Troponin is a protein associated with the muscle contractions. So a basic research tell you that whether the caffeine can improve the rate of muscle contractions. If the caffeine improve the rate of muscle contraction, which is available with a basic science research, so whether I can use caffeine to improve the muscle contractions, or I mean the activity of the muscles contractions, and does it help in the athlete performance, improve the athlete performance? This is how, for example, I gave that this is an example of basic and applied research. So all basic research, they should have some applied aspects sooner or later, maybe in the long run. So coming to a research flow, the research outcome, I have told you that research is a systematic knowledge which is coming to us, or which we get through a systematic body of information and with a number of steps. I must have a critical observation. A researcher should have a critical observation. One should have a rational thinking on the observation. Then I should have a predictive explanation, that we say hypothesis. I must design a sound experiment, well-designed experiment, to prove my hypothesis, to prove my predictive explanation to a particular problem. And once I get the result or the evidence, I must analyze with intellectual ability and profound understanding. And then I should give a findings. These conclusive findings may not be attained with a single experiment. I may need a repeat number of experiments, series of experiments, to come to a conclusion, a conclusive finding. So research is a journey, and the journey may last for few years to few decades. Great discovery never comes easily. So these are some of the steps, as I have told, that how I can plan a study, how I can plan or design a scientific study. So I must find out a problem through my observation, through my information, gathering information from the existing body of the knowledge. Then I should have a hypothesis, then I should do experiment. And once we have a hypothesis, and if I prove to, I mean, able to prove my hypothesis, which is correct, that is maybe converted into thesis, and which may lead to a discovery of a new theorem, or you can say a new idea or new knowledge. Now, if I thought that, yes, I should make science and I should opt for doing science, I should go for a career in science, I have number of options available with me. But one important thing I must require, which should be everlasting throughout your career, throughout your journey, that is the passion. So to keep the scientific temperament alive throughout your journey, you should have a passion for the subject, a deep passion for the subject. Without a deep passion for the subject, you cannot achieve your goal, you cannot make success in your career or professions. Let me give some examples that how passion is the most important driving force, how it is powerful. Let me quote one particular examples, that once I have a passion and the mind is ignited, the ignited mind is the most powerful resource in the universe, on the earth, above the earth, and under the earth. Let me quote one example, and I understand this one example is good enough to convince all of my dear students that how passion is more powerful. So passion is definitely more powerful, and that passion is the most important driving force in our career. let me give you example of the pioneer of our atomic energy program of India, Dr. Homi Jahangir Bhabha. So how passion is a powerful driver to a scientific career. So at the age of 18, he was sent to Cambridge by his parents to train him as an engineer with an ultimate objectives that he was closely associated with Tata group of people, Tata Industries. So his parents sent him to Cambridge to learn engineering, to train him engineering so that he can be the future of this Tata Industries. However, Homi Jahangir Bhabha was not meant for that. He has a passion for the interest not to do engineering, rather a pure science. So I will quote a letter written by Dr. Homi Jahangir Bhabha when he was in Cambridge to his father, that is way back, is 100 years back, you can say in 1928. I quote, I seriously say to you that business or job as an engineer is not the thing for me. It is totally foreign to my nature and radically opposed to my temperament and opinions. I know I'll do great things here, for each man can do best and excel in only that thing of which is passionately fond, in which he believes, as I do, that he has the ability to do it, and he is in fact born and destined to do it. My success will not depend on what A or B think of me. My success will be what I make of my word. Besides, India is not a land where science cannot be carried on. I am burning with a desire to do physics. Please note, my success will be what I make of my work. Besides, India is not a land where science cannot be carried on. 100 years back, Dr. Homi Jahangir Bhabha could thought of this. I am burning with a desire to do physics. Engineering is not my destination. I will and I must do it sometime. It is my only ambition. I have no desire to be a successful man or head of a firm. There are intelligent people who like that and let them do it. So this is, you see, the passion of a scientist, the passion of an individual to pursue his career, and how strong letter, convincing letter is written to his father just to fulfill his passion, to complete his desire. Then when Bhabha could do it as a, and later on we have seen that how Bhabha has cherished his career as an scientist, and I must say that he was the pioneer of atomic energy program of India. And you know that Bhabha Atomic Research Center, that is in Bombay, that is his contribution and his passionate drive. So this is, I have told you, I promised you that one example is good enough to all of you, that how passion is the important driving force to pursue your career towards success. Let me give some example of how the discovery, how this discovery are the outcome of some of the critical observations of our day-to-day life, and with a rational thinking, with a scientific bent of mind. Think of this Archimedes, the Archimedes principle, the principle of buoyancy. So that is from an observation that he could establish that the principle of Archimedes principle, that is only when he was taking a bath in a bathtub. Coming to another example, a great mathematician. Let me give an example, that Ramanujan. So he made a substantial contribution in mathematical analysis, number theory, infinite series, and continued fractions. So let me tell, I think most of you must have heard about Ramanujan, that his ability and how he is an acclaimed mathematician throughout the globe. He could not pass the first examinations in arts, but with his passion to do mathematics, solved number of theorems and problems, and he was later on invited the people throughout the globe to explain his theorem and to contribute. And And that is how I can say that if informal education may not be required, so nothing can be a limitation if you have a passion, if you have a directive thought, and you have a critical observations and a scientific bent of mind. To Ramanujan was once he had told that an equation is nothing to me unless it expresses a thought of God. So science is also being touched or associated with the spirituality, the nature. So we have the natural forces with us, nature with us, and nature is, I can say, the most beautiful architecture, and nature has got all forces, nature has got all invention, so we must learn from the nature. I will tell in my last part of my talk that I must learn everything, we must learn from everything from the nature. I will give another example that all of us, we know Patani Samant is self-designed instrument with the help of bamboo sticks. So he could measure the distance of stars from Earth. And that time he had no instrument, no sophisticated instrument with him. So it is his innovation, it is his desire, it is his passion to do the science, do the astronomy. So my point of telling over here that nothing can be a limitations when we have a strong desire, strong passion to do the things. I can give enough examples, a lot of examples, but we have limitations of the time. So few examples I am just citing just to kindle your curiosity and to inspire you in the right direction to choose science as a career, as a profession with a deep passion. Think of Marie Curie, think of Thomas Edison, the great inventor. Marie Curie, you know, that the first woman to win a Nobel Prize. So it is this dedicated life that how she could solve problems during her lifetime, and she could get Nobel Prize twice. Think of Thomas Edison, who finally invented his great discovery, and once he has quoted that I have not failed with number of efforts, I have just found 10,000 ways that won't work. So doing things, trying things, failure is not, it should not be a dragging force for you. That you can try 100 times, 1,000 times, but there is no harm trying 1,001 times. I will give a very interesting example, very living examples that from my own observation, how critical one should have the observation, and then that leads to a number of questions. All of us were very acquainted with this plant, Basil alba. We know that is a we consume. This is in Odia, it is called pushal. So I have got a seedlings planted in my garden, open garden, grew well. So after 1 month, I was interested to get it to near my kitchen, so it will be in the confined area. So I put it in a gunny bag with some soil. So I lifted the plant and put it in a gunny bag near my kitchen. So after 10-15 days, I could observe that the leaves are getting depigmented. So I could observe that the green color of that leaves, that is being gradually lost. I could not find the reason. I talked to some agriculture officials, happens to be associated with plant pathologists. I discussed with them. They suggested me a few things to spray this thing, that thing. I tried, but nothing could change the things. Simple out of curiosity, I put them back into the garden where from I have got it. After again 10-15 days, I could observe these leaves are becoming again colored. So it is initially that the loss of color, the depigmentation, then again the pigmentation, the lost pigmentation is regained. The only change, only difference is only the soil. The native soil was different, and the soil where I have planted in a gunny bag, that was different. So there is something is there in this soil. So this research is going on. I have discussed with some of these plant physiologists, some of the people, soil biologists, that what may be the reason for such depigmentation, which is very conspicuous, and it could, expressed within 10-15 days. So this is how the beautiful example that how a critical observation is very important to do research. Next, I can give another examples that blending fundamental research with a translational outcome through critical observations and rational thinking. So we do some research in our department in the laboratory, taking different animals as a model, Madam has told, starting from silkworm to rat. So I will give some examples that how we have observed, and following our observations, we have redesigned our experiments, and we could got some of the findings that was not initially we have thought of. So research, I have told, research is not always a linear relation. It's not a linear expectation. So it may give something else which we do not expect, which you do not understand. And so that is a good researcher thought of or peep through a crossroads, a crosstalk, and new things, not necessarily a linear things. So we do research on silkworm, that is, the Anthera mylitta, tropical tussar silkworm from which we get the fabric, clothes. The silkworm, Anthera mylitta, that is a native of India, Bangladesh, and Pakistan, and is very important agriculture crop, you can say, that it is particularly it's a sericulture, and it's a non-mulberry sericulture because it feed on the leaves which are not mulberry. It feeds on arjun, or that is sal, terminalia tomentacea, terminalia arjuna, or asan. These are the three different host plants. And being a student of science, you must know that it is the larva which are reared on the host plant. The larva consumes the leaves, they form the cocoon, is again a protective cover, is a physiological adaptation, you can say. Now the cocoon, that is the source of raw silk, and that cocoon gives to the silk, and the silk gives to fabric. Even not only the fabric, only it is fabric, it has got also a lot of biomedical importance and also a lot of things in the material science. So I will just share a story of this silkworm research, that how the fundamental research, a blended fundamental research, also results in the outcome of a translational product. So this is just to explain you that is the life cycle, and already I have told the larva which reared in the host plant results in the cocoon, and the cocoon the source of the silk. Now, while doing our experiment on this silkworm, we had an objective that since silkworm are reared outdoor, they're exposed to various climatic extremes, temperature, solar radiation, ultraviolet radiations, environmental pollutants. And since we are working, we do work on redox regulations, means that is the balance between the oxygen metabolites, particularly the reactive oxygen species, and a group of molecules of the proteins, we say antioxidants, which opposes the functions of pro-oxidants of the reactive oxygen species to maintain the redox balance. So since we work on redox balance or reactive oxygen metabolism, so we had a bent of mind that maybe that some of the environment in which the silkworms are reared might be inducing, triggering the oxidative stress through the increased production of reactive oxygen species. So what we have designed in our experiment, so how to reduce the level of oxidative stress during the development, which are possibly coming from host plant, possibly coming from environment. So we had taken that one known antioxidant, everybody of us know that is much studied, ascorbic acid, which is a known immune booster. It is also an important antioxidant. So we have used this, we are using this ascorbic acid antioxidant to minimize the possible oxidative stress during the development of silkworm. While we did the experiment, and one of the experimental objective was to study the protein profile, because silk is a protein. So we had some thought of understanding, we have some understanding. So silk is an induced protein. So ascorbic acid or other antioxidants which are using as a supplement in through the leaf might be inducing some protein to be upregulated or downregulated. So one of the experimental objective was to study the protein profile in response to ascorbic acid supplementation. So we did simple SDS space, and we could found the particular protein, particular molecular weight of the protein, around 75 kilodalton protein is being overexpressed in a particular tissue. We are dealing with three different tissues, like hemolymph, which is more likely your blood of your vertebrate, and fat body, more likely your liver of a vertebrate, and the midgut, which is more like your stomach. So three tissue are working to understand the redox regulatory mechanism in response to different antioxidants. So to our, at the end of our experiment, we could observe a particular protein. So we focus our study to understand this nature of the protein, that what is to reveal the identity of the protein, and what is the possible role, the function of this protein. As a part of our next experiment, we try to identify the protein. And since SDS space has the limitations that it cannot reveal the identity of the protein, we went to the next experiment. We observed this particular protein is expressed highly overexpressed in a particular tissue, in a particular developmental stage, in response to a particular supplementation. So we focused on this particular protein band, and we moved to our next experiment. We tried to understand that what this protein may be. Another important observation, why I am talking these things, that observation is very important. So we observed that this particular protein, 75 kilodalton protein, is highly expressed in hemolymph of this insect. And very interestingly, in the hemolymph of the same insect, we didn't find activity of an important antioxidant enzyme, that is your catalase. So catalase is an antioxidant enzyme which neutralizes hydrogen peroxide, and it neutralizes hydrogen peroxide, by which that it also reduces the chance of developing oxidative stress, because too much of hydrogen peroxide along with iron may lead to production of this one important radical, hydroxyl radicals, that leads to oxidative stress. So catalase functions to minimize or to quench or to neutralize the amount of hydrogen peroxide, excess amount of hydrogen peroxide. That is how it restrains the system, or that restrain the tissue from the development of oxidative stress, particularly iron mediated oxidative stress. So we had two observations. One is this particular enzyme is absent in this particular tissue, that is hemolymph. In contrast, that same hemolymph express a particular protein which is not expressed in that much of high concentration, the high level in other tissues. So we thought that there must be some relations, there must be some compensatory role or functions. Maybe during the course of evolution, it is a evolutionary driven phenomenon. So when we understand that catalase is absent, and in contrast that particular protein is expressed, we were more interested to identify the protein. Next, we moved to an experiment, that how to identify the protein. Then we tried to identify the protein through the mass spectroscopy. So we excised this particular band, went for the in-gel digestion, and then we identified this protein through the mass spectroscopy. Though the facility was not available with us, we moved to other place and got it identified. And on identifications of this particular protein, which is induced in response to ascorbic acid, not only in response to ascorbic acid, later on also we observe that particular protein is also expressed as such inherently in a particular developmental stage of this larva. And that particular protein is nothing than transferrin. So transferrin is a protein, the name suggests has been studied in other insects, has been studied in many organisms, including human being and mammals, and very important protein associated with iron homeostasis, associated with iron transport. That gave you a good thought process in our mind. So what is that? So catalase is absent. We could not find the activity of the catalase in this tissue, which is destined to, one of the enzyme destined to neutralize hydrogen peroxide and also restrain iron dependent oxidative stress by minimizing the level of hydrogen peroxide. Another important things we observe in our another study, that hydrogen peroxide is an important signaling molecule in this particular insect, which regulates its dormancy and the process of aging. So with all those hypotheses, we develop a new hypothesis. So possibly is an evolutionary driven phenomenon. Hydrogen peroxide is essential for this organism to maintain some of the physiological function, but catalase is absent in that particular tissue. So hydrogen peroxide balance need to be maintained in a particular threshold level so that the tissue should not develop oxidative stress. So it is maybe nature or the evolution has selected this particular protein, this transferrin, and which can minimize the hydrogen peroxide driven oxidative stress, not by sequestering hydrogen peroxide, rather by sequestering iron. So it's a different mechanism we proposed, and we could publish these things in a journal. And before that, we also tried to evidence, or we tried to be more clarified that yes, this particular protein may be associated with maintaining oxidative stress. We did some additional experiment to give some more evidences. Yes, this particular protein is associated with maintaining the oxidative balance or redox balance in this organism. So we developed a hypothesis that hydrogen peroxide is essential for this organism, but whatever the reason, we do not understand properly that hydrogen peroxide scavenging enzymes or regulating enzyme is absent in this particular tissue, and instead of that system biology or the system nature has given another protein to minimize the level of oxidative stress, which is a combined contribution of iron and hydrogen peroxide. And this we could give a new hypothesis. So we could give a new role, we could explain the new role of transferrin in this insect, though transferrin was known as an important component of other insects or mammals. So this was the first report from our laboratory to explain, yes, transferrin has got an important role in the regulation of redox regulation. So we didn't stop here. So when we went to identify this protein through mass spectroscopy, and that you understand that mass spectroscopy is a very sophisticated device, not every laboratory can afford, and a lot of money is also required to run the mass spec to maintain the mass spec. So if we want to identify this protein all the time, we need a mass spectroscopy facility. So what we thought, that why we cannot develop an antibody against this protein, so that instead of doing mass spectroscopy all the time, we can detect the protein by the immunodetection, either by Western blotting or by ELISA or by immunohistochemistry. So we were interested to develop the antibody, of course, the polyclonal antibody against this protein. And this antibody was successfully raised, and not only it is only functional to detect this particular protein in number of tissues in the insect, even in the other insects, including the Drosophila, but that protein, that antibody has also been commercialized. That Avigenex India has happily taken and has commercialized. Now it is available in the open market, and you can take this, buy this, or you can use this to detect the transferrin, not only this insect, also in other insects, including the very important model organism that is Drosophila. So this is what I have try to share these things with all of you, that research is not always a linear relation. It's not a linear expectation. So anything you can expect out of your experiment, but you should have a critical observation, you should have a rational thinking, and you should have a knowledge, background knowledge to analyze the things. That is what I say, intellectual knowledge and the profound analysis. This is the story of this generation of new knowledge and development of the product, which already have told is the integral part of science, doing science. So later on, that when the story is going on, this we have got the transferrin could develop the polyclonal antibody during this COVID-19. I mean, this pandemic situations. I could find out some papers that those where it is it is available in the public domain that transferrin is also a potential molecule contributed to COVID-19 severity. So we got excited that yes, the molecule which you have discovered, which you have reported for the first time from this insect, has the ability not only to do the physiological functions in the iron metabolism in this insect, so it may have a first role in something contributing or something controlling COVID-19. The research is on, research is undergoing. That one to with our antibody, we have now trying to detect this particular transferrin in mammalian system, and once we'll able to do it, we can use this antibody whether the increased level of transferrin can be sequestered by giving an antibody therapy or something like that. So, but it is not an immediate translational output. It may be long term, it may take years, but it is maybe definitely an avenue. We do not know, one day may come. This can be a potential therapeutic molecule. It is already established potential therapeutic molecule in other diseases like cancer and neurodegeneration, but we need a lot of study to establish our hypothesis. Next, I will come to another story. So this is about the blending, the fundamental research with translational outcome, the story of remodeling redox regulatory network with the transferrin, as we reported for the first time in this insect. Coming to this, another story I wish to share, just to explain you that how nurture is important. Nature and nurture, if I say that one is our genetics, another is the epigenetics. So when sometimes genetics has the limitations to explain something, the window of epigenetics is open. So epigenetics is nothing which is over and above the genetics. Genetics is nothing which is the linear sequence of the nucleic acid, the DNA. So when the linear sequence of nucleic acid DNA, which is translated into the protein, ultimately fails to explain the epigenome. When the genome fails to explain the epigenome, the epiproteome comes into picture. That is what I say, that is nature versus nurture. So when nature is nurtured in a different environment, product is different. So when your talent is nurtured, when your talent is being value added in the right kind of platform and right kind of intervention, the product is different. So this is just to briefly explain you that genome and epigenome, there are different type of epigenomic modifications. One of the important type of epigenomic modifications is the methylations of DNA. DNA, that particular base that most often the cytosine gets methylated, means the 5 carbon of the cytosines, the one methyl group is added, and when methyl group is added, it becomes 5 methyl cytosine. Also sometimes it becomes 5 hydroxymethyl cytosine. So when the DNA gets methylated, means methyl group is added, it behaves in a different. So when a DNA is methylated, it exhibits a different type of response. It particularly it leads to an epigenetic regulatory effect. Epigenetic regulatory effect means there is no change in the sequence of DNA. Without change in the sequence of the DNA, when a particular base is being methylated, this is one of the example of epigenetic regulations. besides others. I am not going to discuss the limitations of the time. So when the DNA get methylated, that regulation, the genetic regulation is something different. That I'm going to share few findings story, also story from our department, just to explain you that how epigenetic is a powerful driving force as a part of genetic regulations. So this is an example, so long back, that how this methylation is associated with the body color. See, when a pregnant rat was fed with a specific diet supplemented with a methyl donor and fed with a regular mouse diet, the body color was different. Body color was different. Not only body color is different, the susceptibility to different disease is also different, and also the lifespan. So when a simple diet has the ability to influence not only the body color, the disease, it can decide the disease susceptibility, it can determine your lifespan. So if that particular supplement which has the ability to induce methylation has got do lot of things in your body physiology determining your lifespan. I can give another example. Not only that feet, even the tactile stimuli, the touch, body touch. So you can see that this is a very important experiment done by Smini, Weaver, and Skype. It's a very remarkable experiment. It gives a lot of idea on epigenomics, particularly the DNA methylations, that good mothering versus bad mothering. A good mother rat licks and groom her pups, so gives them extra space to suckle against their underside. Bad mothering rarely bare licks her pups and provide almost no tactile stimulation. So this is the difference. That is how the grooming, that a good mothering means that how the maternal care has the influence in the last part or the rest part of your life. So this is what epigenetics can explain. Genetics cannot explain, because the same genetic makeup, only the difference in the diet, difference in the environment, different in the rearing conditions, even the different in the emotional touch may lead to the different type of phenotypes, different types of physiological manifestations. And here that one group of pups are stressed and the group of pups are not anxious one, rather relaxed one. So this is what epigenetics is. Next one, I will give some example by taking very important molecule we use in our day-to-day life. That is curcumin. It's a wonder molecule abundantly found in our everyday life, abundantly used in our everyday life as a part of our cooking process. That is your turmeric, haldi. So turmeric contains a good amount of curcumin, and that curcumin is also an important epigenetic modulator. And I will share a few experiments, 1 or 2 experiments, to explain how this curcumin can be an epigenetic, can prove as an epigenetic modulator. So we do some work on also thyroid disorders. You know that thyroid disorders is one of the important metabolic disorders, endocrine disorders throughout the world, including also for India and our state. 1 in every 10 Indians have some kind of thyroid dysfunctions, and we are doing research on thyroid disorders since a long time. So our objective was to study that thyroid dysfunctions or thyroid disorders associated other pathological manifestations, say oxidative stress, inflammatory response, how they can be regulated, because thyroid disorder can be managed by taking a drug destined to treat either hypothyroidism or hyperthyroidism. Both hyper means excess of thyroid functions and hypo, less thyroid functions, are harmful to our metabolic system and a metabolic threat to other different pathological conditions, including hypertension, being associated with the diabetes, also very remotely associated with the kidney functions and neurodegeneration. So our objective was that how curcumin intervention, means curcumin can be used for the better management of, as an alternative therapy, or not alternative, is an adjunct therapy to treat the hypothyroid or hypothyroid patient, taking rat as a model system. So in the taking rat as a model system, that we have injected PTU or given PTU through their drinking water. PTU, the propyl thiouracil, is a drug, and it is also a drug used to induce hypothyroidism in experimental conditions. So when we feed them or allow to drink them with PTU, they develop hypothyroid conditions. Under these hypothyroid conditions, that ROS level is elevated, they develop oxidative stress, and that oxidative stress mediated pathogenesis in the long run. So these thyroid dysfunctions, when they results in other pathological conditions, we have intervened with curcumin. And since liver is one of the important target organ which is being affected during altered thyroid status, we have used liver. Besides, also we have used the testes and kidney and other organs also. But we have done a very important epigenetic study taking liver as an organ, and we have observed that most of the molecules or the proteins which regulate these epigenetic machineries, that DNA methylations, they're mostly the group of proteins or the enzymes which are associated with the maintaining the methylations and indirectly demethylations, they are being altered, either sometimes upregulated or downregulated. This is not only that the machinery, that epigenetic machinery is upregulated or downregulated. Earlier we had an observation that during the process of these thyroid alterations, that catalase protein or the catalase enzyme is being downregulated. Means catalase activity is being compromised in some tissues. And so we try to understand through epigenetic process that what may be the possible mechanism, that how come this catalase is being downregulated when there is hypothyroidism or when there is the intervention with propyl thiouracil. So we could observe that the promoter regulating the expression of the catalase is being altered, is being methylated, hypermethylated due to this PTU treatment. And curcumin to some extent that ameliorate these conditions. Coming to the another story. So this is one story about this, how the curcumin can be a good epigenetic modulator, and particularly taking the example of thyroid alterations. Coming to another important findings, that since we are working on curcumin, and when there was the COVID-19 pandemic, so we thought of, my research scholars, our research group, we thought of that if the curcumin has got a wide range of ability to interact with the different proteins, thousands of proteins in our body, so can it interact with angiotensin converting enzyme ACE2, that is the important gateway through which that coronavirus entered to human cell. And we did some computational study, we got some findings, and that is under, we're in the process of, publishing this in a very reputed journal, and hope these findings will also contribute us to understand that how not only curcumin along with another important molecule abundantly found in the green tea, catechins, that they are able to intervene the binding of this SC2 and this H protein. Coming to an indirect evidence, coming back to the Silkworm story, we did some experiments, since we know that we have some information that survivability of the silkworm was variable in response to different host plant. It has got 3 preferred host plant, this Arjun, Asan and Sal, that is Arjuna, Terminalia, Arjuna, Tomentis, and Robusta. So we did an experiment. We did this experiment in-house. We tried to rear this larvae in the laboratory, feed them with the different plant leaves, and we could unravel the fact that the information that the larvae feed on Soria robusta has a very low survivality rate. And we could understand, we could find out from our study by analyzing of the leaf by a very sensitive method that particular induced X-ray emissions, that this particular leaf has got good amount of iron. And iron is a very important inducer of oxidative stress. And we could observe when the larvae were fed on this leaf, particularly the Terminalia tomentissa, they have elevated oxidative stress, and possibly this elevated oxidative stress can be a cause for decrease the survivability and mortality. Further research is under process to give further evidence, more direct evidence, whether this is really because of this elevated oxidative stress or some other synergistic physiological phenomena exist. Then a very important observations on again this with the silkworm. You know that insect has a fascinating world, that insect has a very important stage in their developmental period, that we say diapause, a pause in development, a temporary stoppage in their developmental stage. And you won't believe that this particular silkworm species, the Antheromylata, they have a varied lifespan. The same strain of silkworm having the same genetic makeup, in one season it exhibit the lifespan, the pupal lifespan is 21 days, and another season it exhibit an elevated increased lifespans, maybe you can say 7 to 8 weeks or sometimes more. So how this such variation in their lifespan occurs? So how do they age into this different, same population, but only two different cycle of life, two different life cycle? How the process of aging is so drastic, how the process of development is so drastic, and which triggers this dormancy. So they have a period of dormancy, what we say, that is the diapause. And how come the period of dormancy in one case it is 21 days, in other that is more? We could find some interesting results. We could correlate that it is again possibly the redox balance, and particularly the level of hydrogen peroxide, already have told, is a very important physiological molecule. Also in the insect, it's a small molecule, but it has a very big role in biology, a very important role in biology. So possibly this hydrogen peroxide and this level of differential level of redox balance may be a determining factor in the determining the lifespan and also regulating the dormancy. Other people throughout the globe are also actively working on this particular phenomenon, that particularly in insect, how the dormancy is regulated. Even they have got some interesting results that how epigenetics play important role, and possibly that is regulated through the differential regulation of some set of heat response or cold response gene. So these are the some of the story that already I shared, just to inspire my dear students to make your career is very fascinating in the field of science. And the thing that is again very fascinating, the insect polyphenism. So same insect has got different type of morphological characters. Again, it is also being regulated by the diet and also by epigenetic mechanism. Though we have not done study on this, but information, as per the information available with us, the insect polyphenism means the different morphological features of the same individuals are associated with the DNA methylation, which is a very important type of epigenetic modulator. So this is what I wanted to share, that how the passion is a very important driving force, and research is an integral part of doing science, and how the research can give you and the findings we do not expect. Sometimes it gets unexpected result, sometimes we get expected result, but research is always full of unexpectations, full of uncertainty. But you must have a reproducibility to sustain your research. So out of all the stories which I shared so far, to choose science as a career with a number of avenues for the professions, and to do everything, for everything you need a strong passion, and that is must be supported by a critical observation and a rational thinking. Now, if I go back to this, so my message to all of you, there should not be saturation in your passion. A time will come, a career is saturated, your profession is established, but your passion should be ever growing. And if you have an ever growing, everlasting passion, I don't think your career is never saturated. You will have an uplift in your career, and you will get utmost satisfaction in your profession. So passion, the major driving force for everything doing in a science, then your career, then your profession. So to do scientific research, sustain scientific research, which I have told, that leads to innovation, lead to transformation of the universe. Your sustained scientific research with deep passion gives to technology development, technology refinement, and sustainable, eco-friendly, and economically viable technology, inclusive technology, that is the technology for the society, for the societal transformation. So my message to sum up in the next few minutes, we have heard from some of our previous speakers, and I have tried to summarize everything in my lectures. I am fortunate that I gave lecture later on. I could have the opportunity to learn all of the speakers, including my teacher, Professor Jivian Chaini. And my essence of telling this, that I have told nature is great, nature is beautiful, Mother Nature, that is our natural force. Learn from nature with deep passion, search and research within nature. Research is a noble profession. When you do science passionately, your career and professions are assured integral parts. So if you do science passionately, you don't have to think for your career, for your professions. So now you may get the answer whether you do science for career, for profession, or for passion. And for everything to do, you are blessed with, you are gifted with a sophisticated think tank, that is your crazy brain, which can give the answer for everything in the universe. Every question you ask, this crazy brain can give you the answer, can give you the direction to move further. My last slide. Brain is the real laboratory. That is none other than Jagdish Chandra Bose has told. And this is what I mean to say, what I wish to give my message to all my dear students, particularly the new generations, the forthcoming people who do wish to take their career in science. The transformation is the outcome of a first-sighted vision, innovative and guiding spirit. And with this, I come to end of my lecture. So I have a brain, I have a right brain and left brain, and balance of everything. And my passion is coming from the right brain. My reasoning, my analytical ability, my aptitude, my attitude to science, all those things possibly coming from the left brain. So the purpose is the reason for your journey. Passion is the fire that lights your way. And sometimes your journey during your research career, the momentous mobility, the journey is more meaningful than your destination. I gratefully acknowledge the contributions, inputs of all my teachers, students, scholars, and colleagues in this journey of research in Utkal University before that. And with this, I must express my gratitude to the organizers, giving me an opportunity to share my thought, my experience out of my journey in academics, teaching, and research. And whenever I have got an opportunity to do, I have tried my level best to do the things with a deep passion, and possibly that is my driving force. And I never repent that doing science, coming to a profession of science, I have come out of my choice. It's not never a chance. I have never thought of doing science, other than any other professions, only doing science. Thank you. Thank you all.
Thank you so much, sir, for the very interesting talk. We are really delighted. I, along with all the participants, would like to thank you once again, sir. It was a really enlightening topic, I would say, giving us the insight into how to do the research, what should be the aptitude of the students. And as far as passion is concerned, I conclude that the passion is directly proportional to our satisfaction at some stage of life. So thank you, sir, for inspiring the students with all such practical examples. We have a few questions, sir. Can we take a few?
Yes, please.
Sir. Mr. Bishnu Prasad Sahu wants to know if, sir, could you please bring light upon how students can choose from classical field of science and applied field of science? What kind of skill or mindset is required for one to succeed in classical field of science, and what are the requirements for one to pursue applied field of science?
Okay, so it's very nice, interesting questions. So I've told that whether it is a classical field of science or applied field, already I've given an example that difference between the basic and applied research. So research is research, whether you do a basic research or applied research. You should have a bent of mind, research bent of mind. You should have enough curiosity, and mostly the sustained research leads to a findings which is applied, leads to a technology. So I don't think that you have a specific bent of mind to pursue in fundamental research and to pursue in applied research. So research, whether you do fundamental, foundational or applied, that you need the same level of your interest, same level of inquisitiveness, and most importantly, same level of your passion. The fundamental research may some more time to give a complete conclusive findings. But we must remember, when we have a findings out of our fundamental research, that is translated into a applied research. So fundamental research is also equally important, and translational research is no way less important because it is directly for the benefit of the society and the mankind. So I don't think there should not be any difference aptitude for doing science, whether it is fundamental or applied. You should have the same level of aptitude, but sometimes a little difference in your approach maybe. Because when you think of the translational research, you should think that how you should have a value-added products. So that is what is a technology refinement. So when the outcome is coming, that is the raw materials, and there is the possibility, there is the scope that further improvement of that technology, further improvement of that product, that is what we say value additions. So value addition is definitely very important to define your technology and to define your product. I can give you an example that I have heard from a great man long back, that the India has the vast reserves of minerals, resources. So most of the minerals, including the ores for iron, are being transported to Western world. They have robust technology. They are technologically sound than us. So with their robust and I can say improved technology, they give the value-added products, and all those value-added products we import. So we are having the raw materials, maybe because of lack of appropriate technology and ability for value addition, they are being transported, means exported, and now then we are being imported. All those things are imported. So when we go for a translational research, we should have all those things with us, that how this product should be marketed, how the product should be of utmost benefit of the society for the humankind. Hope I could answer the question.
I hope the same, sir. There's one more query. A student wants to know, how does choosing a model for an experiment alters the course of action? And he wants to know specifically if it is advantageous to go for an invertebrate model or a vertebrate model organism.
Can you please repeat the question, please?
How does choosing a model organism alter the path of research?
Oh, yes.
He wants to know if it is beneficial to work on invertebrate groups or vertebrate groups.
You see that every organism has got its own limitations. Every organism has got its own advantage also. So suppose I will say that I want to study the effect of a particular drug, how this drug is purposeful or useful for human being. So in that case, I will take more phylogenetically related organisms which is more close to human beings. Phylogenetically, not necessarily, it may not be phylogenetically regulated, but at least its genome should be almost similar, or it should be, minimum difference between the genome and the biochemical pathway. You see that most of the biochemical pathways, except few, they are basically conserved. The basic mechanism conserved, depending on the time during which they have evolved in the earth, availability of resources, availability of the food, and the way they're nurtured. What I have told you, they have been modified. So whether it is the vertebrate or invertebrate, that model need to be very robust so that less variability will be there and your reproducibility should be there in the repeated times. So whenever perform the experiments 3 times, 4 times, that repeated results should be reproducible so that you can come to a conclusion. But I don't think that you see that Drosophila is a very important model. Now zebrafish is becoming an important model. And sometimes the alternative model, people choosing the alternative model, then your mammals, because of various reasons. Maybe one of the reasons is your stringent principle of your animal ethics committee, availability of the animals, less sacrifice of animals. Even we have a cell culture model. But cell culture model is something different, which is a totally different, because it's not the system biology approach. So you can take any system biology with a similar physiological mechanism that can give you the right answer, and you can always compare this with, to give a more comprehensive findings, you can compare with more than one model also.
I hope that would have answered the query of the student, sir. Thank you once again. May I now request Mitali Sahu Madam, Faculty, Department of Geology, Case Deemed to be University, to kindly present the vote of thanks. Madam, please.
Thank you, madam. A very good evening, everyone. Respected and most distinguished guest of the day, Professor Jageshwar Dandapat, Head PG Department of Biotechnology, Banibhar. Respected all my dear colleagues and all participants. It seems to be a great honor to propose the vote of thanks to all who have helped us in making this webinar a very resounding success. First of all, I would like to give my hearty thanks to our distinguished speaker, Professor Jageshwar Dandapat, for making an excellent presentation and making this webinar very meaningful and interesting. And I am very happy to express a vote of thanks to our
Just a moment. We are having a bit of technical snag. Just a moment.
start. Very good evening, everyone. Our respected and most distinguished guest of the day, Professor Jageshwar Dandapat, Head PG Department of Biotechnology, Banibhar. Respected all my dear colleagues and all participants. It seems to be a great honor.
Just a moment. Just a moment. We are having a bit of technical snag over here, sir. I apologize. I apologize. We'll be back in a moment, sir.
So this is yet another aspect of research, having lots of patience, dealing with all kind of technical snags, and adjusting with the situation. So we are back again. Madam, please.
Okay, thank you, ma'am. It seems to be a great honor to propose the vote of thanks to all who have helped us in making this webinar a very resounding success. First of all, I would like to give my hearty thanks to our distinguished speaker, Dr. Jageshwar Dandapat, for making an excellent presentation and making this webinar very meaningful and interesting. I'm happy to express a vote of thanks to our colleagues that has made our webinar grand success. Then I would like to give my vote of thanking the coordinators, organizers for making this webinar a great success. Finally, I thank the wonderful students and all participants. Thank you so much for your cooperation. Once again, I thank all for your cordial cooperation. Thank you so much, sir. Thank you.
Thank you, madam. Thank you, sir. Thank you, everyone.
Thank you, everyone.